So in the new drug development part, you would have the toxicology being studied in context to first in human studies, FIH studies. So how does it signify, how does it affect, and how regulatory toxicology is of very utmost importance in terms of understanding clinical trials is what we'll be looking at now going forward. So here you have the initial testing in humans. That's the first in human. That's the one which has been basic trial phases which you're aware of in human beings. So initial testing in humans is being done. Normal volunteers or patients with or at risk for the target subjects are checked out. Open label dose escalating safety and tolerance pharmacokinetic studies are being conducted. Healthy volunteers and patients are being looked into. Healthy volunteers for the phase 1 trials and patients for the phase 2 onwards. Intense level of monitoring is being done, metabolic studies point of view. Flexibility in making the dosage adjustments is being done because we go from microdose trial to the major dose, to the normal dose trials. Formulations have been worked around, so that is all what we'll be talking into. While healthy volunteers and patients, I had last time also emphasized on the fact that in case of trials pertaining to neuropsychiatry and oncology, two therapeutic areas, you do not have healthy volunteers there. It's always patients being there because the medicines do have an effect very different from the normal other medicines which you talk about. So in two, neuropsychiatry and oncology cancer trials, we do not work on healthy volunteers as phase 1 trials. Okay, guys. So now to give an influx of these activities, how it is significant in terms of video format, so I'm going to take you to a video point. So here I'll share you about clinical trial phases. So from T4 onwards I'll be showing out. Guys, any trouble regarding sound or video? Are you able to see it clearly? Because I have shared the windows only, the desktop only directly. So I'm just asking, is it fine or
Yes, sir. We can see you.
you can see and you can hear it also, beta? There's a sound also.
Sir, can you play again?
Sir, can you play it?
Yeah, I'll play. Is it fine?
So
No,
there's
sir,
no.
there is no sound.
So there is no sound. Okay, one second, beta. Let me share it again. Then open share tray, include system video, desktop. Okay, I'll play again now, guys. Just confirm, and then we can go ahead with that. All right.
Think about it. The fact that you're alive and watching this video really is quite amazing.
Can you hear the sound also now, beta?
Yes, sir.
Yes, sir, yes.
Oh, oh,
Yes,
okay,
sir. We
okay,
can hear.
okay, okay. Oh, fine. That's fine.
Every minute of every day, you're being exposed to potentially lethal chemicals. And I'm not just talking about the man-made stuff here. Oxygen, water, carbon dioxide, the chemicals that are in your food. Get too much of them inside you and you have a problem. To make matters worse, our bodies are constantly making and using incredibly potent chemicals as they go about their daily business. Our cells and organs are in a constant frenzy of activity as vital but potentially lethal chemicals are inhaled, ingested, created, processed, and eventually got rid of. Of course, this is the very essence of what it is to be alive. Cut out the chemicals and the seemingly crazy mad processes that they're part of and you'll be, well, dead. To stay alive and thrive, our bodies pull off an incredible balancing act between making use of the stuff we breathe and eat and are otherwise exposed to, and making sure it doesn't kill us. The trouble is, it's a balancing act that can too easily be disrupted. Too too much or too little of something, and the whole system begins to get out of whack. Because of this, staying healthy sometimes takes more than good intentions over what we put into our bodies and what we don't. It depends on knowing something about the science of how substances interact with our bodies and potentially cause harm. And this is where toxicology comes in. Toxicologists study what happens to stuff when it gets into your body, where it goes, what it does, and how it leaves. They investigate the harm that can be caused when too much or too little stuff interferes with your internal balance, and they help devise ways to avoid harm or to fight back when things get messed up. Not to put too fine a point on it, but toxicologists are the Sherlocks of the health world, using science and evidence to understand and protect our bodies down to the very DNA that makes us what we are. Toxicologists know that we can't escape from potentially harmful chemicals. We need them to remain healthy and to ensure the environment we live in is safe and good for us. But they also know that if enough of the wrong stuff gets to the wrong place at the wrong time, things can get messed up fast. As a result, this is a powerful science because it gives us the key to staying alive and thriving in a world that could otherwise be an awfully dangerous place. So whether you're wondering if your shampoo is as good for your body as it is for your hair, or you're concerned about what's in your veggie burger and fries, or even if you're trying to work out how much chocolate a reasonable person can eat, a toxicologist might just turn out to be your best friend.
So the major emphasis of this was what toxicology is all about. And now we will see a quick video of clinical trials. So you know how it makes a significance here.
Clinical trials are research studies that involve people. Most clinical trials to test new cancer treatments are done in a series of steps called phases. If a new treatment is successful in one phase, it moves to the next phase. Let's take a look at the three main phases of clinical trials. Phase 1 trials are small, enrolling less than 30 patients. Phase 1 trials are designed to find a safe dose of the new treatment, determine how the treatment should be given, and learn how it affects the body. If a safe dose is found, the new treatment goes to Phase 2 testing. More patients are enrolled, usually 100 or less. Phase 2 trials study how the treatment affects the body and how the treatment works for a certain type of cancer. If a new treatment is found to be safe and to have some benefit, it goes to Phase 3. This is when many participants are needed, usually more than 100 and sometimes thousands. Phase 3 trials compare the new treatment with the current treatment to see which one is better. One thing is certain with any phase trial. By taking part, you will help future patients by helping today's scientists learn more about cancer. For more information about clinical trials, including their possible risks and benefits for you, go to cancer.gov/clinical trials.
So guys, this gives you an idea in terms of what are the three major phases which you have: phase 1, phase 2, and phase 3 clinical trials. So we understand that toxicology is important. We understand that the phase of clinical trials are important because these are giving us information which is very significant to us. So there has to be regulatory part of controlling both the phenomenons and regulatory part influencing both the things. So we will go into that details in the next video, but before that, do you have any questions guys as of now? Because next video is but a little bit complex in the sense we are going to be listening to what FDA's perspective is all about, and I'm there to explain what the inference is to be made out from the entire video section point of view. It will be giving you a perspective of regulatory toxicology, of what regulatory toxicology actually infers out from a regulatory perspective, where these two phenomena are looked into together and especially for the special populations like pediatrics, geriatrics, oncologies, and quality of life enhancers. All these are affected by the phenomenon. So we are gonna be looking into next video, which is all of it's a little longer video, and it's gonna be talking about FDS perspective. So FDS perspective in that is what is gonna be going ahead, and you can ask me to stop in between if you have some questions. I'll explain that thing. Otherwise, during the video, I'll be stopping at many places and to explain out the concepts to you. Okay, guys. So I'm continuing with that, believing that you are able to see and hear it very clearly.
So you want to know more about toxicology? Well, toxicology, also called...
Sorry, guys. This is another video which has got popped up. One sec, guys. Oh.
Good afternoon.
Okay, guys, you can hear it. This is Terry J. Miller
Yes.
from US FDA talking about it. Pitfalls in preclinical development from regulatory perspective in terms of toxicology, regulatory toxicology application pertaining to clinical trials as such. Okay, so this is going to be 18-minute video, and we'll be stopping in between as Terry goes on telling, and I'll keep on explaining.
My name is Terry Miller, and I'm the Pharmacology Toxicology Supervisor in the Division of Anti-Infective Products at the FDA. I'd first like to thank the organizers of this conference and our moderator, Ursula, for the invitation to come speak with you. Today, I plan to provide a regulatory perspective on the challenges or pitfalls typically encountered in a review of the non-clinical developmental programs. I would like to begin my presentation with an introduction into non-clinical development from a toxicology perspective, discuss the current farm tox requirements for a successful, streamlined, and non-clinical program, provide examples of non-clinical issues encountered in early and late development, and conclude with some overall recommendations. As you may know, the drug review process is a complex, multidisciplinary, stepwise process involving evaluation of animal and human safety and efficacy data. The Farm Tox Reviewer assesses the non-clinical safety data submitted by the sponsors. Efficacy data, particularly in animal models of disease, are reviewed by other disciplines on the review teams. The focus of the toxicology review is to provide hazard identification with recommendations on clinical monitoring. In our assessment of non-clinical studies to support first-in-human dosing, we typically ask ourselves the following questions. Is the non-clinical approach reasonable? Is the first-in-human starting dose safe? What are the most likely toxicities expected in the clinic based on animal findings? Are these toxicities reversible? And has the sponsor identified an optimal dosing schedule and study duration in animals that adequately supports the clinical trials?
So, beta, this is the first perspective of what FDA looks into. So, FDA is looking into these 5 distinct questions while they are reviewing a particular documentation from the sponsor, which is giving them an idea about whether the sponsor is fully ready and has done the homework enough to go ahead and do something called first in human studies or phase 1 clinical trials. The first is the non-clinical approach reasonable? So what it means is the in vitro studies, is the in situ studies, is the studies in animal 3 species novel concept studies approach correct for that particular intervention to be attributably justified for use in humans? later from the dosing point. Is the first in human starting dose safe, and has it been microdose? Microdose means what happened is, supposingly, if you are giving a particular drug in a certain proportion to animals. So in animals, what you do is you give the drug in a proportion by milligram per kg, or that's weight by weight or weight by volume, anything of that sort. And when you want to give it to the human beings, you give 1/100th of that particular dose which was given down to, which was found in animals to be lethal. So what are doing is you're adjusting the dose in such a level that it is not toxic to human beings at any cost. So what are the most likely toxicities which have been observed is what they will be looking into. And then they will also want to know is the toxicity related or not related. That means you stop the drug and does the effects go away, or does it continue out? So if it goes away, that means it can be monitored, controlled, and based on the dosages. What is an optimal dosing schedule, and how do you arrive it out based for the supporting clinical trials? So these are the 5 parameters which they look in context to any of the entity which is been going down to them.
The good thing is that there are many non-clinical guidances to help advise you on your non-clinical development programs. The principal guidances are the ICH guidances that were negotiated and agreed upon by several international regulatory agencies, including the FDA. The most important non-clinical guidance bolded is ICH M3R2, non-clinical safety studies for the conduct of clinical trials, which describes the type, duration, and timing of non-clinical studies. In determining the non-clinical data that you can rely on for your submissions, we generally make the distinction between the investigational new drug application and the new drug application or marketing application. For the IND, you may rely on studies that you conduct, published literature and published databases, the summary basis of approval for other FDA-approved products, and drug monographs. However, these sources must contain sufficient details to base an independent safety decision. For marketing applications, you may rely on any studies you conduct and published literature, FDA labels for other products, which we will review for adequacy. The FDA summary basis of approvals and any other FDA advisory committee materials and non-USA regulatory assessments cannot be relied upon. Full reports of investigations of safety are required to be submitted in the NDA for marketing approval. Overall, a good non-clinical package at the time of NDA submission would consist of a pharmacodynamic assessment, safety pharmacology studies, single and repeat dose toxicology studies, genetic and reproductive toxicology studies, carcinogenicity assessments if they are deemed necessary, and any special toxicity studies such as.
So guys, this is the list of studies which is expected from a non-clinical study perspective. As part of investigational new drug application, you must have submitted this data, or as a part of sponsor or a CRO or whichever organization you will be working, you will be submitting the data. So the data will be pharmacodynamics in terms of in vitro and in vivo, whatever you have done. Safety pharmacology with special emphasis on these 3 organ systems, okay, or you call it as system organ classes. One is central nervous system based ones. Second is respiratory, that is respiratory system based ones. Third is the cardiovascular intervention based ones. So these are the 3 primarily looked out safety pharmacology, organ study informations or system organ classes informations being there. The general toxicity is based on either single toxicity studies or repeated dose toxicity studies, whatever has been attributably submitted. Genetic toxicity influences out the pharmacogenetic perspectives and how it has been genetically shown to be, because there are certain chemicals which are oncogenic triggers, which can trigger genetic mutation levels to high level, and that's what you call as mutagens. So if you are using the mutagenic components, then the aspect of genetic toxicity is very important there. The reproductive toxicity is very important from the based on the fact that thalidomide was one of the safest molecule, but it was found to be reproductively toxic. That means it was found to be affecting the newborns. So that's the reason it has been important to understand the reproductive toxicity. Carcinogenicity studies, normally you have aims test to just have teratogenicity and carcinogenicity of the molecules being studied out at the initial levels only. And that's what is being used as carcinogenicity study details being there. The special toxicity studies is including the immunotoxicity, that means the body, the immune system based ones, where you will have the pharmacodynamic actions being exhibited very clearly, giving you the immunological parameters. And phototoxicity, which we are talking about, where we had last time discussed out, like smog, environmental pollution point of view. Okay, there can be compounds which could be effectively affecting at the phototoxic levels. So those are the ones which are called special studies of phototoxicity and immunotoxicity studies. So all pivotal non-clinical studies which are being conducted has to be aligned with or in lieu of GLP, good laboratory practices. It has to be GLP certified ones, and this has to be a versionation which has to be justified for their studying part of it. So these are the recommended non-clinical study information which is required as per FDA phenomenon or FDA's perspective looking at it. Is this clear to you guys? Any questions here?
No, sir.
Okay, brother.
No,
Thank you so much. Okay, going further.
Immunotoxicity of phototoxicity assessments. It should be noted, with the exception of the pharmacodynamic studies, all pivotal non-clinical studies should be conducted GLP. You should keep in mind that not all of these studies need to be completed before your first-in-human trial. As described in the ICH guidance M3R2, there are certain studies that are required at each stage of clinical development before the next stage of clinical development can be initiated. For example, prior to the first-in-human phase 1 trial, we typically would like to see a safety pharmacology core battery, single and or repeat dose toxicity studies in at least 2 species, rodent and non-rodent, in vitro gene tox assessments, tissue cross-reactivity studies for biologic products, and qualification of any novel excipients in a clinical formulation. Prior to start of phase 2, for example, additional repeat dose toxicology studies that better match the intended clinical duration and exposure, if changed, may be needed, along with an in vivo genetic toxicology assessment to round out the gene tox core battery. Prior to phase 3, fertility studies and embryo fetal development studies in at least 2 species, rodent and non-rodent, and any juvenile animal studies are typically recommended, as well as a discussion of the impurities and chemistry issues with the final products. Also by this time,
So, but when we are mentioning, when Terry is mentioning about impurities and so chemical manufacturing control issues, that means yield parameters. So every active principle which you are talking about as, okay, you call it as new chemical entity, which further becomes prodrug and drug, has to be as pure as possible, has to be looked in from the yield perspective of a chemical reaction. That means it is producing enough yield of it, and the salt basis of it, so sodium salt or potassium salt or what sort of a salt it is all about, and the impurities which are associated with that has to be taken into account here. At the same time, its cross reactivity with other compounds would have been studied and would have been reported. At least whatever has been available is what is required before the start of the phase 3 trials. That's what has been mentioned out here in impurities and CMC dose part.
completion of the PK-ADME assessments in animals should be submitted for review. And finally, at the time of NDA submission, the peripostnatal development toxicity study should be completed, as well as any carcinogenicity studies, if needed. Carcinogenicity studies for drugs to treat serious and life-threatening illnesses may be accepted post-marketing, but we ask that the studies be initiated at the time of NDA submission. As mentioned, the most pivotal non-clinical studies required to support early clinical trials are the single and repeat dose animal toxicology studies. In these studies, we typically look for the following. Are the single dose or multiple dose studies needed to support the clinical trial? Are the planned treatment durations and anticipated clinical exposures covered in these animal studies? What are the target organs for toxicities in animals? And is there a dose response for these effects? And what is the maximum tolerated dose, the no observed adverse effect level, and no observed effect level values identified in these studies? And most importantly, are these effects reversible?
So, these are the general considerations as I used to tell you, and I have been telling you from the start of this particular presentation about regulatory toxicology from last couple of lectures. Is that it has to be single dose or multiple dose studies, and you have to be very sure about the treatment durations and the clinical exposures covered. That means treatment durations in the sense what we have planned out and we have seen the design part of the trials. When we say design part of the trials, we know about treatment durations, how long the drug has been administered, how much concentration, plasma concentration the drug is all about, and clinical exposures does make a significant entity there, right? Then what are the target organs? So we had studied this in the way how the drugs are absorbed, the pharmacokinetics and pharmacodynamic part of it, how the spread occurs, the one compartment or two compartment systems. That is what we are talking about in target organs. Then is there a dose response for toxicity effect? So you have to know about the very specific nature of the dose response curve pertaining to that particular drug, whether it's a prodrug or the new chemical entity which is now moving towards to become a drug. Then we should also be knowing about the MTD, maximum tolerated dose. Okay, and then I always have been mentioning about NOEL. NOEL is this one, beta. No observed adverse effect level. NOAEL, NOEL. Okay, in 3 animal species, 1 non-rodent and 2 rodents. That's the way how it is being there. So rodents and non-rodents both are taken into account in that. And the most important question, is the effect, whatever we seen, reversible or non-reversible? Okay, because that's very significant, because if it is non-reversible, it will add on to toxicity because it's accumulating in one of the organs or the compartment systems. So when you give the later dose, it's just going to add on to it, and that chemical will cross the adverse event level for that particular organism, and it will be very difficult for treatment perspectives. So these are the things which are looked into general consideration for toxicology studies, which you always looked into. And this now you know is coming from the FDA directly. So this is from Terry, who is speaking from FDA.
For biologic products, toxicity studies should be conducted in 2 pharmacologically relevant species. If no relevant species exists, then transgenic animals or homologous proteins should be considered. The biologic products we most typically see in our division are monoclonal antibodies directed against foreign targets, say a bacterial toxin. And in these cases, a short-term toxicity study in a single species, preferably a rodent, is acceptable. No reproductive toxicity studies are required. For all biologic products, gene tox and carcinogenicity studies, generally, these studies are not needed for biologic products. In the animal studies, you should characterize any antibody response to determine if these responses are neutralizing to the product. You should also consider any PK or ADME differences in animals that may require a change in dose frequency and treatment duration in animals. While juvenile animal studies may help address pediatric concerns not addressed by clinical.
So this was the reason, beta, why we had looked into pediatric. Pediatric we had seen as a separate slide deck only. I had explained in one of the lectures the pediatric part of it, the pediatric toxicity point of view, because it's pediatric toxicity is one of the prime concerns which they have, and they want to differentiate it from the normal levels and then want to know about it. So this is the pediatric toxicity which we are saying. They are also called as juvenile toxicity studies.
Or tox studies in adult animals. The need for such studies to support a pediatric indication is determined on a case-by-case basis. You should plan to provide scientific justification for why they are or are not needed. Juvenile studies are typically conducted in one species, preferably rodent. Non-rodent species may be appropriate with scientific justification. In your studies, consider the indication, the age, the extent and timing of dosing, and any PK/PD differences, the corresponding developmental stages, and the specific target organs identified in the adult toxicity studies. There is an increased risk of toxicity to organ systems that undergo considerable postnatal growth, and toxicity in these organs are typically the reason why such studies might be recommended. The need and timing of combination toxicology studies are dependent on the availability of safety data for each product. Combination toxicology studies are typically conducted in a single relevant species of equal duration to the clinical trial, not to exceed 90 days. It is generally considered that for any two drugs intended for use only in combination, or when the non-clinical data is lacking for each drug alone, combination tox studies should be conducted prior to the clinical trials. Similarly, two early-stage drugs or drugs with a low margin of safety, similar targets of toxicity, or when the adverse effects are difficult to monitor in the clinic, the combination studies should be done early in development. For early-stage entities with clinical experience, combined with late-stage entities, or when two late-stage entities with no combination experience are combined, combination toxicity studies can be delayed until later in development. In general, combination tox studies are recommended for all later-stage trials, longer duration trials, and for marketed combination products, with the exception of two late-stage or approved drugs with prior clinical combination experience.
So special consideration is given in terms of combination toxicology studies, where why is it required and why it is being said. So it's very simple part. For example, we have a single relevant species giving you the idea about the toxicity and the specificity of the drug, and which is correlatable or correlating enough with the human beings being there. So it's only one species, then we will have to run for more than 90 days because we would want to know it's a prolonged time point. While the trials are going on, we should have at least completed 90 days of that particular organism. So correlatively, it will giving you an idea about safety profile of that particular drug. However, if it is more than one relevant species which is already in part, so you may probably will be able to take it account for about 60 days, 65 days, that's fine, but you will get an idea about it more better. Because the systems are different, though, so, and it has similarity with human beings, so simulation study information is considered to be okay and relevant till that time. So you are asked to keep on reporting the IND levels, the safety data of the studies which is going on in terms of animal studies while the clinical trials have been permitted and you are into the clinical trials, the phase 1, phase 2, phase 3, whichever is going on. So this is the one which is very important, starting from the example of, as I said, the glitazone molecules which was found to be inducing pancreatic cancer while it was being long-term studied in the rodents, and it was found that in the long-term usage, the glitazone molecules, specific glitazone molecules, would lead to pancreatic cancer. That becomes more serious as compared to treating the treatment of that particular molecule for diabetes, because if you are at one time controlling the sugar levels, but at the same time the molecule is going to be triggering out cancerous condition or pancreatitis or pancreatic cancer, the later part, which is the most serious non-treatable type of cancer, then the risk benefit is not in the proportion of benefit point of view, it's more towards the risk point of view, so you have to drop the drug. And that is one of the reasons that one of the molecules, pioglitazone, has been dropped off from the stages, and it was a good marketed drug being there, but its later stage study, which is an IND of preclinical ones, which is continuing further while the clinical trial has been completed, marketing authorization provided, and pharmacovigilance was being observed out. That's the time it was determined or detected that it was causing pancreatic cancer in a long-term study, not in the short-term study, not the short-term study of 90 days, but it was getting to have being seen In the rodent population, over about a third year of their introduction of the drug into the system. So third year in rodent. So it was attributably approximately after 5 to 8 years of human usage. So that's the one. That's the reason why these trials are very important, and that's what is the combination toxicology studies which he has been talking about. So I believe this is clear till here, Beta. Any questions as of now?
Sir.
Okay, Beta. Thank you.
There are many common obstacles encountered during our non-clinical assessments that can derail or delay a program. These include incorrect types and timing of non-clinical studies to support the clinical trial, when the no observed adverse effect level of target organs are not correctly identified in the animal studies, and when the length of the non-clinical studies does not match or exceed the intended clinical use. When pivotal studies are not GLP, and when there are issues related to changes in the formulation late in development that results in precipitation or changes in the PK/ADME or toxicity profile. Also, sometimes the toxicity of the excipients alone or the inactive ingredients have not been adequately assessed. This may occur with new or novel excipients or when there's a new route of administration. We typically ask that in these cases, the final clinical formulation be tested in the non-clinical studies by the same route and duration as proposed in humans. Another common problem is when impurities are not adequately qualified in the animal studies. This might occur when batches used in the non-clinical studies fails to contain the impurity in question. Insufficient reproductive tox batteries could sometimes prevent initiation of phase 3 trials and NDA approval. Because carcinogenicity testing can take 2 years to complete, their need should be addressed early in development. I would now like to provide some examples of early and late non-clinical issues that in our estimation slowed a clinical program. First case was for a new molecular entity drug for a chronic indication. The drug caused mortality in several dogs treated for 14 days consecutively and was associated with cardiac defects and seizures. Histopathology of the brain and heart proved inconclusive. However, there were significant QTc effects noted. The drug was shown to accumulate in the heart, brain, and lungs at 5 times greater than plasma levels. The sponsor was placed on full clinical hold and was required to conduct a tissue distribution study and a 90-day dog toxicology study, which they were able to identify a no observed effect level for both toxicities. The sponsor was allowed to proceed with their trial, and several healthy volunteers immediately developed CNS adverse effects after just a few doses, and product development was discontinued. In the next example, also this product was an NME, was a novel protein targeted to a bacterium. 4 weekly IV doses to dogs caused inflammation, decreased blood pressure, and reflex tachycardia after the 3rd or 4th dose. The animals showed evidence of a type 1 hypersensitivity or anaphylactic response. Also, 7-day rat and dog toxicology studies with a 3-week recovery period showed adventitial inflammation, increased heart and lung weights, and damage to arterioles, particularly in the lung. It was suspected that both type 1 and type 2 hypersensitivity reactions were occurring in these animals.
So here the examples are given, Beta. The case 2 is an example of a biological molecule because as you can see, it's a novel protein. So it can be a biological molecule as currently you must be hearing about vaccines being there. The vaccines trials are also having same sort of phenomena. So anything in terms of biological molecule, it goes down to the CBER division, that is Center for Biological Evaluation and Research division of the FDA only. So here again, you can see the influxes, what you can make out from how the study went across. 4 IV doses to dogs cause inflammation and reflex tachycardia. So the rhythms were being affected, heart rhythms have been affected, and it was showing type 1 hypersensitivity. So another one is other than dogs, you have the rat and dog toxicology. Rat was also being there as a rodent, and dog was also being used out. That means it's a combination trial which you're talking about, the one which we just heard about beforehand. And its duration is day into 7 and then 3 week recovery period time point. And it was showing adventitious inflammation. That means inflammation of the heart muscles, aorta, pulmonary trunk, arteries, vascular necrosis. So this particular entity, novel protein, was playing around with the effect on the cardiac muscles. Okay, to give you the slight example right away, for example, in terms of the mRNA of COVID-19 vaccine has been shown to have facial paralysis in cases, okay, Bell's palsy. So what it means is when you are integrating a particular molecule into your system, it may sensitize some tissue uh, epitopes, or there can be some part where it can be sensitized to and it can incite a reaction or a response. Increased heart and lung weights, that's what it means. Another one is said, so obviously it is increasing the heart and lung weight. That means the protein is interacting very well with the heart, cardiac, and the lung, so pulmonary regions. Damage to arterioles, especially in the lungs, was indicated that it is obviously detrimental for the cases pertaining to cardiovascular and cardiorespiratory systems. Suspected was first and third hypersensitivity level reactions, and that is the reason why I require additional animal sensitization study for hypersensitivity to be approved of. This is case 2 of what Terry has mentioned. So anything which you are having a doubt here, Bida, please let me know. Otherwise, we can move on further. Is everything clear, guys? Do you have any doubts?
No, sir.
Okay. Okay, Bida. Thank you so much.
The sponsor is placed on full clinical hold and required to conduct an animal sensitization study. The third case was for a topical gel formulation of an approved drug with a new route of administration for a 5-day treatment of full-thickness burn wounds. Preliminary studies in affected animals with full-thickness wounds limited to less than 7 days showed evidence of efficacy and early evidence of healing. However, these animal studies used different excipients than in the clinical formulation, and the local skin toxicity and wound healing was not adequately assessed. It should be noted that the systemic toxicity of the active pharmaceutical ingredient was not a concern, as the API was approved for oral administration. The sponsor was placed on full clinical hold and was required to conduct a GLP study in an uninfected full-thickness burn wound model with the final clinical formulation administered topically to cover the full clinical duration of treatment.
So here is a case number 3 where the formulation is a different one. This is a skin-based one where you are treating the burn wounds, full thickness burn wounds have been taken treatment out. So in the treatment of full thickness burn wounds, we had an oral antibiotic for a 5-day treatment. It was being understood, it was good. So oral was very well approved out. So the API is active. But when it was being given as a topical gel, that means you change the formulation, you have to do a study. DAB point of view, you need to do a study. So this formulation when applied on the skin would have different effects. So preliminary studies in infected animals with full thickness burns showed reduction in bacteria, reduced inflammation, etc., and early evidence of healing. But using when you use different excipients for as an excipient, because here the change was the point was API along with now you have excipients which are being used. So excipients may be say DMSO, or it may be something else which may have been there, or gelatin based. Some of the excipient, as I said, was probably cross reacting and was showing an effect on the skin other than the normal one which is being there. That means this now is to be studied, because what was the effect of that particular excipient with the API as a formulation of a gel administration. So the gel which was now being made had to be requested by the FDA, as you can see, for an uninfected study. So in an uninfected case, is the skin cells being sensitized because of the gel application? Is what was they wanted to know before they wanted to approve this particular molecule. So you can see these are the case studies which these are the cases which you can say as their examples. Their final report says how they found that to be very different in these three scenarios. So this is a case 3 which has been mentioned by Terry.
I would now like to describe 3 cases where non-clinical deficiencies were noted late in development and impacted the NDA submission and review. The first case was for a new molecular entity, oral and IV drug to treat an acute infection. The sponsor needed to rely on older pivotal animal studies conducted decades ago to qualify for several product impurities. It was determined that the impurity profiles in these non-clinical lots tested in these early studies were not adequately characterized. And also there was marked emesis noted or vomiting noted in the dogs throughout the study, likely affected the plasma levels.
So this was a late development case. For example, when we say late development case, we are talking about NDA, new drug application. So it had crossed the IND barrier. Now they want, for the marketing point of view, prior to marketing point of view, they wanted to have a check on it. So the prior to marketing point of view, when they have the NDA submissions, a new entity which is being used out, so they were being checked for one part. The sponsor had mentioned in the NDA about the trials which they had done for IND levels, for animal studies, which was very old pivotal studies. So very old pivotal studies they had talked about, and the only thing which they had mentioned was emesis. Emesis means vomiting by the dogs when they were being given this particular drug entity for acute infection. So when you say emetic, that means the drug is not reaching the plasma level and it is an oral drug. So that means the plasma concentration levels are not going higher. So you can't be very sure about the toxicology of it, okay? Because the body has a mechanism by which whatever if you have eaten, if you have not been very good with it, it is toxic to your body. The body tries to throw it out. So when body is trying to throw it out, and it is showing an emetic behavior, obviously the concentration levels are not gonna reach what you must be targeting. So that can't be looked at from a safety perspective. So hence this was being told for additional 28-day rat toxicology studies by the FDA group. So you can see these cases gives you an idea about how and where you may encounter a particular time point where the FDA would want you to go ahead and conduct further studies as part of sponsor. You may not think that way, but they have their own valid reasons why they would be asking for more studies in certain parts. So these are examples of that. So this is a late development case which has been mentioned by Terry.
Sponsor was then required to conduct an additional 28-day rat toxicology study with enriched impurity levels to qualify these impurities before they submitted their NDA. The second example was for a new molecular entity drug for acute infection that involved a late formulation change just prior to phase 3 and long after the non-clinical studies were completed. The late formulation change included a new excipient known to increase absorption of some drugs up to 50-fold. The sponsor conducted a 2-week IV bridging toxicology study in rats which showed no differences in PK or toxicity profile. It was noted, however, that the reproductive toxicology studies were conducted with the prior formulation. The sponsor is required to conduct an additional fetal distribution study in pregnant animals with the new formulation, as well as an additional embryo-fetal development study prior to submitting their application.
So this is another one of a new medical entity or new molecule being there in picture, in development at the NDA stage. What they did was they wanted to change the formulation at the phase 3 level. So a phase 3 level is going on and they want to change the formulation. They showed the IND report with the study of rats where they showed a bridging toxicology study with no difference in pharmacokinetic or toxicology profile in the animals. So that was with sufficient reporting point of view. However, there is a question. What about the study pertaining to, because after thalidomide there is a skepticism in the regulatory safety part. So what exactly? Okay, fine, this is fine, but where is the data for reproductive toxicology? So you can see the FDA had asked for fetal tissue distribution study in pregnant animals also before approving it, so that they know that it is not toxic to the fetal level, or the reproductive toxicity is being also taken care of if the formulation change has occurred. And that gives an idea about how EFD toxicology study is a requirement for a new formulation, for a new approval, and that's developmental case 2, which has been presented by Terry.
The third and final example was for an oral antibiotic to treat a chronic parasitic infection. The standard battery of non-clinical studies were submitted to support NDA approval. It was noted that the reproductive toxicology studies conducted back in the 1970s lacked evaluation of several key developmental milestones in animals. Therefore, the applicant was required to conduct a reproductive toxicology study in alignment with current standards defined in the ICH guidances.
So here is another example, Beta. For example, this is a question where you are talking about an oral antibiotic drug, okay, or antimicrobial drug for chronic parasitic infection which is being there, and what Terry is mentioning is about the reproductive toxicology study which was done in 1970s was used as an inference for providing that this was a safe drug. But when you say in 1970, there have been a lot of studies which was not done. For example, reproductive toxicology was not taken care of in 1970s at the earlier time point. So what it means is the studies were not completely relevant as of now to have supported the findings or supported the phenomena which they were targeting out. Hence, the FDA, in spite of having all the reports being complete for an NDA approval, new drug application approval, or marketing permissions or marketing approval and authorization, asked the sponsors to provide additional reproductive toxicology studies, which should be in line with the current standards for post-marketing point of view. So that's the way how it is being done. So there you can see this is a case 3 of late development. So you are seeing how the FDA looks into from the regulatory toxicology and asks for study details in terms of basic BABE studies specifically pertaining to regulatory toxicology to ensure that the drug is approved out and is being marketed out, is being permitted to market, and that's what and where you have the significance of regulatory toxicology comes in picture while being clinical trial approvals have been taking place. So is this clear to you guys? Any doubts here?
Yes, sir.
Great, fantastic.
In this case, we did agree to accept the study post-marketing because of the indication. However, had we not agreed to this late submission, the need for this study could have impacted marketing approval. In my overall recommendations, I would like to mention that non-clinical drug development is a complex process guided by ICH and FDA guidances and prior division experience with similar products. Because of this, the process cannot be streamlined with appropriate guidance from the division and by toxicology consultants with knowledge of the process. We highly recommend you seek advice on your non-clinical developmental approach as early as possible. We suggest a pre-IND consultation with the division, but do come ready to discuss your non-clinical approach and any data that you may have. We are willing to review pivotal animal study protocols before you initiate your studies, but do provide us with at least 6 weeks to review and comment on the protocols. I sincerely appreciate your time and attention today. And again, I would like to thank Ursula and the Eshmid organizers for the invitation to come speak with you. I provided my email address as well as contact information in the division for whom you can address any questions that you may have.
So this is from the FDA. So you are understanding the regulatory toxicologies and its influences in direct context to how the FDA looks at it and how it has been significant to understand the regulatory toxicology. That means toxicology in perspective to regulations being looked out by regulatory bodies. So now, after having an idea about this, we can come back to our original point where we were looking into the perspectives of clinical trial testing and how regulatory looks at it and how it's important for dosage and regimens being there. So let's go to the next slide. As you can see here, so I've just changed the slides here. I'm moved from video to the slides now. So this phase 1 and clinical pharmacology studies, how much is the cost? It's about 1.5 billion per drug. This is something a little bit older one data. As of now, what we have is about approximately 2 to 3 billion per drug. And that's a huge cost which is involved in this particular studies. So this, it has discovery component, it has preclinical component, and preclinical component gives rise to the help for IND, investigation new drug. Then you have the phase 1, phase 2 and phase 3 trials. Then you have something called as submission of NDA, as you can see, and that's an approval, and then you can market it out. That's called as MAA, marketing authorization and approval. So in the 21st century, what are the trials which have been normally looked into? So in the preclinical part, you may have microsomal trials, you may have calcium carbonate based ones, or permeability ones, solubility ones, alginate ones,
Hello,
entrapment
sir.
studies, enzymatic profiles, etc. Yes, beta?
So we can't see the presentation. Have you started it?
Presentation, beta, I have kept a slide on. Can you see the slide?
No, no, sir. We are
Oh,
still on the video.
सर
you are still on the
स्लाइड
video.
दिख रहा है फेज 1 एंड क्लिनिकल फार्माकोलॉजी स्टडी का
हाँ बेटा, वो स्लाइड दिख, अच्छा सबको नहीं दिख रहा है बेटा, कोई बात नहीं, मैं एक बार और शेयर करता हूँ. वन सेक. थैंक यू बेटा फॉर टेलिंग मी. ओके, दिखना तो चाहिए क्योंकि बेटा इस बार डेस्कटॉप मैंने शेयर किया है. आई शेयर इट अगेन. कैन यू सी इट नाउ?
यस सर
Yes.
Okay, now you can see. Fine. Good. So I was telling about this slides beta. Okay, move from the slide because I had shared the desktop, so I was just thinking. No problems. It happens like that. It's technology, and it can happen to anything, and everything can happen. For example, Google also had an outage in the last 2 days, so it's okay to have technological challenges. It works out. It can be worked out to the better. Okay, so when we talk about the human volunteer PK profiles, these are the trials which are being there, patient PK profiles, PKPD models and simulation, the PK studies and IVIVC modules. These are the ones which are being done. Another ones are the drug interaction, disease states, special POPs and BE studies which have been done to ensure that we are very thorough with the IND and the FDA does not have questions pertaining to it. So next one is, I moved to the next slide, beta. This slide heading is the regulations and a few guidance documents is what is being said. Okay, hope you can see that. So the first one is from the 21 CFR, that is Code of Federal Regulations as per FDA 320. That's the number which you have to check out for. ICH guidance documents are the 15 plus S1 series onwards is what you normally want to look into. EMA guidelines, the documents are about 30 plus guidance documents for pertaining to this FDA regulatory toxicology point of view. General BABE guidance you have to check. Population PK guidances you have to check. Exposure response guidances you have to check. Then Animal Welfare Act, USDA regulations you have to check. Good laboratory practice you have to check. The BCS guidance, the IVIVC guidance and SUPAC guidance. These are about the infrared modified with the multiple resonance parts and the single cell store studies. All these have to be checked out, and these are the guidance documents and regulations which you check out while going ahead with the trials. So the general BABE guidance is intended to provide recommendation to sponsors planning to include BABE information for orally administered drug products in INDs, NDAs, and ANDAs. Okay. So are also generally applicable to non-orally administered drugs. Products one relies on systemic exposure. Also, you require the BAB study. So we had seen one of the cases which was mentioned by Terry, where you could see that formulation change was the only thing which was being done, and then FDA had asked out for reproductive toxicology details for approval. So that happens. That keeps on happening. Another one is PKPD implication in a new formulation or new dose of approved drugs, where we check out for blood levels. The pharmacokinetic data alone is not the only one which is looked into. We always look into PKPD and the efficacy trials, and nowadays it's more emphatically looked into teratogenicity, carcinogenicity, and reproductive toxicity. So these are the three terms which you have to remember: teratogenicity, carcinogenicity, and reproductive toxicity, which are the three important parameters which FDA and the EMA looks out before approval of IND or NDA. Then you have the FDAMA, which talks about 115 section, which says clinical investigations support one adequate and well-controlled clinical investigation with a confirmatory evidence, which is confirmed out from the metabolic profile of the organisms as well as the first in human studies. That you call it as PK and PK over PD studies. So PKPD, why bother? Well, FDAMA, that is approvals, section 115, clinical investigations, states congressional committee reports, which says confirmatory evidence is scientifically sound data from any investigation in the NDA that provides substantiation as to the safety and effectiveness of the new drug. So this is what is being emphasized on. Confirmatory evidence is consisting of earlier clinical trials, pharmacokinetic data, or other appropriate scientific studies. So these are two parameters which they are looking into. So FDA Modernization Act, that is FDAMA, what we have said is an act of 1997, and section 115 was what we had referred to. Section 111 is for pediatric study of drugs. So where you have to have PK bridging studies reports to mention about the pediatric population as a necessity. So FDAMA is Modernizing Act of 1997 beta. You have to remember that section 115 and section 111 are important. In the pharmacokinetics, you have to look into 21 CFR part 201.56, which is all about the pediatric labeling regulations, which says FDA may approve a drug for pediatric use based on studies in adults and other information supporting pediatric use. Additional information supporting the pediatric use must ordinarily include data of pharmacokinetics of the drug in the pediatric population, or otherwise the other information such as data on the pharmacodynamic studies should have been mentioned in the label. So the labeling perspective is also an important part. So during the process, how does it work? So in a clinical trial process, one of the most important thing is attrition on the R&D process. For example, average about 7,000 to 8,000 compounds are screened based on the system or the class of compounds, okay, based on functional group entities, of which, so for example, 7 lakhs maybe compounds may be screened, of which you will come down to about, based on preclinical pharmacology, you come down to about 100 discoveries. So you can see the narrowing down. From 7 lakh or maybe more than that, you come down to narrow down to 100. Of the 100, you have lead compounds. Of the lead compounds, you come down to maybe 10. From the 10, we will move down to maybe 1 or 2 at the end. So when you do the preclinical pharmacology, clinical safety, clinical pharmacology and safety, from the 7 lakh plus compounds, which you must have on an average searched out into 7,000 on would have been what have been from the high throughput screening methodologies, you come down to, in a span of 11 to 15 years, as low as 1 or 2 molecules, which goes up to full development. So it's a very high attrition which is observed in drug development process, and that's the way how it is. So what does this process include? One is changing the industry point of view, target validation, screening, lead development, pre-lead, lead, first in human. The first in human FIH is after an IND. Proof of concept, FDA dialogue, full development. That's after phase 3. You have an NDA. So this is the way how it works out. So when you look into these studies, how would you explain them as? First is exploratory IND, then second is microdosing, then is PKPD studies, then is biomarker-based studies, desired human exposure studies, proof of non-viability studies, proof of concept studies, and finally approval. That's the way how the procedures move into. So critical path agenda and opportunities. What happens is critical path is, that means accelerated approval requirements for studies. You have a very valid example as of now here. Normally vaccine trials run for an approximate time of 10 years on an average, nothing less than that. 10 years is an average. Now this is the first time when the industry has been able to work around for a vaccine development for COVID-19 in about approximately 10 months time. So when you can say 10 years based of work is what normally you spend across to get a particular vaccine out. And today for COVID, it is about 10 months which people have spent out. Okay. So you can see it's called accelerated approval. It's called critical path agenda and opportunities. It occurs because of the need being very high and the time and the mortality rate being very high, leading to less time to develop and bring about something which is very significant, drives this process. Then that's called as critical path agenda. It does have for various diseases. For example, there is a critical path agenda for AIDS. There's a critical path agenda for Ebola. Nothing has been done as of now for Ebola. Then there's a critical path agenda for the mysterious X. That's one of the viruses which is being known to you. Cause and now the critical path was for COVID-19. So COVID-19 is vaccine development, at least for the part which has been critical path development part. So model-based drug development approaches are powerful tools to improve clinical drug development and regulatory guidance and quality of NDA submissions. So model-based drug development such as drug and disease modeling, exposure response modeling, these are the ones which are being used out across industry point of view. And me being in industry can vouch for the fact that we are increasingly using artificial intelligence in clinical research to get across these two modeling aspects, that is drug disease modeling, exposure response modeling. We have been using it very instrumentally to bring about this particular change, and artificial intelligence has been helpful in simulating molecular mimicries and QSAR to such an extent that we have been able to understand these details very well. So that's what is an important tool, beta. So in today's arena, you do not only work on the labs, you use QSAR. Guys, by the way, are you aware of QSAR? What is QSAR, beta? Hello? Guys, you around? QSAR, beta?
No, sir, no.
What is QSAR, beta? Any idea?
No idea, sir.
Okay, beta. Okay. So, beta, in a typical laboratory format, what we do is we find out a chemical, we cross-react that chemical with another chemical. Normally, we can write down the reaction based on what your understanding is, based on the functional groups. And then we would go ahead and do that sort of experiment in the lab under normal temperature and pressure, NTP and parameters to determine what is the compound formed. Now this takes a long time to build up and bring about actually what happens. So there is a software system which is called Quantum Structure Activity Relationship, QSAR, Quantum Structure Activity Relationship, which is used as a high throughput mechanism to bring about the chemical molecular mimicry and parameters to explain these concepts. Wait a sec, if I have a video, I can just show it to you what exactly QSAR is all about. I do have a video on QSAR. Okay guys, can you see the screen with the building a QSAR? Hello guys, please confirm beta if you can see the screen with
No, sir.
No, sir.
no. Okay,
No,
I'll
sir.
just okay. I'll represent it to you. Wait, guys. Include system audio, desktop. Okay, can you see now a black
Yes,
screen
sir.
with building a QSR written down on it? Okay.
Yes, sir. Yes, sir.
Okay, beta. This is a small video section which I am gonna show it to you. It's about 6 minute and 43 seconds odd. It's gonna give you an idea about how QSAR is used nowadays. The traditional one of doing the reaction in the laboratory, spending time, and those. Other than that, what they do is they simulate everything on screen, and you have the QSAR giving you the analysis of what is the bond length also of the molecule. What we do is we spatially orient the molecule and we dock it. So for example, when you say two molecules are reacting with each other, that means the functional groups are docking each other, is what is the concept which is occurring out. So we are able to do this in a three-dimensional space in a simulated way in this software system, and which gives us the idea of what is happening and what sort of molecular formation occurs. Because we give the energy levels of each atom and each bond length. So what happens is it gives you exact spatial orientation of the molecule and the reaction chemistry of it. So what happens is then you cut it short and you are able to deliver the molecular response or molecular functionality faster without actually performing an experiment. And then what happens is once you know this particular thing, how it's being working out and how the things are being done, we can go ahead and now perform an experiment in the lab to confirm that this is what is happening, what was being predicted by the QSAR. Now this was what was being done in 2004 onwards. This software system is available. 2000 it was available. 2004 the highest version came out at that time point. We have used it, so I have used it personally, so I am aware of the entire thing. KM Kundani College of Pharmacy is where I had used out that particular software. The person whom I had worked with is his name is Dr. Anwar Sheikh. He is in Alana College in Pune. He's HOD there. So we both had worked together on this particular aspect for bismuth subsatellite set for our molecular study. So it has been available from quite some time, beta. So now what is the next level is this QSAR, which we used to look into, is been looked upon by artificial intelligence, AI. So the machine learning artificial intelligence package looks into it. So as we used to think about the probabilities, the machine calculates the probabilities with the deep root technology, and it brings in molecules which are similar of that sort of a chemical composition, which we would have otherwise thought from our brain point of view, ideas would have occurred to us, and then we would have moved that further. So what has happened is entire process has been cut down from years to seconds now. So earlier an entire scientist used to spend about 15 years, 20 years to develop something. The same thing can be analog by analog techniques and use of QSAR and AI. It can be done in approximately 2 hours. So I'm just showing you this particular video in that point of QSAR, and then you can think about how AI works, machine learning point of view. So we'll just understand that part also later. So playing for you now.
Step, you'll probably notice that the interface has a lot in common with the previous hypothesis scoring step. Near the top of the interface, we have the hypothesis table. In this case, populated only by the selected hypothesis or hypotheses that we chose to carry forward. And at the bottom of the interface, we have the ligand table and the ability to keep ligands in the workspace as we switch between hypotheses.
So here, beta, this window, what you can see here, this window is indicative of hypothesis. Hypothesis means what we are saying is, okay, supposingly if you add, say, an acid and a base, salt is created. Okay, so acid plus base gives you salt. So you are hypothesizing that you have an acid and that's here. Now you have bases, so you have probable bases list here. So what you say is, okay, acid plus base gives you salt. So that's your hypothesis. So what sort of acid at what level will give you a base or will give you a salt is what is you are thinking in your mind, and based on the chemical structural interface of that particular compound, this particular software will calculate it out and give it to you. Okay, so that is your hypothesis and this is your ligands.
Just like in the previous step, we also have the ability to export hypotheses, save them to Maestro's project table, or delete them altogether. You can also set up excluded volumes from within this step as well, where you might wish to use the three-dimensional QSAR to help guide the placement of excluded volumes. That said, there are some unique features in the QSAR step, and as you've probably already guessed, these features all have to do with QSAR building and visualization. At the top of the interface, you can see that there's a tab that allows us to switch from the hypothesis table to a table of QSAR results. We haven't yet built a QSAR, so the table is empty right now. What I want to do now is to go ahead and build a QSAR, and in the process of doing so, we'll take a look at some of the other features in this step. To get started, we'll click on the Options button, which brings up a window that allows us to adjust some settings during the actual QSAR calculation. One of the important options here is the seed value. Right now, the seed value is set to 0. With a seed of 0, every time we separate our ligands into training and test sets, we'll get a different result. If we set the seed to a positive integer, we'll get a random result, but it will be the same each time we perform the separation. Let me show you what I mean. With the seed value set to 10, I'll click Apply here at the bottom of the Build QSAR interface, and 50% of the ligands are assigned to the training set used to build a QSAR, while the other 50% are in the test set that's used to evaluate QSAR model quality. Clicking the Apply button again doesn't change it because I'm using the same non-zero seed as before. I still have the same random selection. Now let's see what happens when I set the seed value back to 0. As you can see, with a seed value of 0, I get a new random selection each time. Before we go ahead with building the QSAR though, I want to show you a few other options that you can set. These options allow us to specify the grid spacing used when building the QSAR, the maximum number of PLS factors to use, and whether or not to eliminate variables based on a specified t-value. That last option will allow you to eliminate variables that are overly sensitive to small changes in the training set composition, so let's go ahead and enable it. The model type option is quite important. Here, you're telling Phase whether you want the QSAR to be based on the placement of atom types or the placement of pharmacophore features. For atom-based QSARs, Phase will use the placement of atom types in space to predict ligand activity. For pharmacophore-based QSARs, Phase will predict activity based on the placement of feature types with respect to the hypothesis. In this example, we'll go with the default option and create an atom-based QSAR. If we were to have created a pharmacophore-based QSAR, we could have specified the effective feature sizes in this table below. Let's go ahead and actually build the QSAR model now by closing the Options window, clicking on the Build Models button, and submitting the job. We're only building a QSAR for a single hypothesis, so this is a pretty quick calculation. One thing that you'll want to note is that if you didn't align the so-called non-model ligands during the previous score hypothesis step, Phase will take care of this prerequisite automatically when you run the QSAR calculation. When the results are incorporated, we can click on the QSAR Results tab and take a look at our results. If we were to have more than one QSAR in this table, you'd see that it works a lot like the hypothesis table that we're already familiar with. The first thing we'll do is to visualize our hypothesis using the toolbar at the top of the interface. The size of each of these cubes you see reflects the earlier setting we saw, wherein we could specify of all atoms.
Hello guys, can you hear me? I
Yes,
thought
sir.
we got disconnected in between. Okay, beta. Okay, I'm sharing the tray again. One sec, guys. I believe we got lost here. Okay, can you see the screens, guys, now?
Yes, sir.
Okay, beta. All right, so I'll play it out for you now. So what we were going through was this model. So you can see the structure being there. So you can see when the person selected one particular chemical entity and one ligand to react out, you could see it is giving you the dimensions, the data pertaining to each bond length, bond value, which is being there between R2 RL levels and R10 levels. So you can see it gives you a structure and the possibility of what new molecule could be made. Now that's what is QSR all about. So playing it further for you,
types in the QSAR. However, we can zero in on the individual effect of a single atom type very easily. We'll do that by clicking on the QSAR visualization button. The QSAR visualization interface is divided into two parts. At the top of the interface, we can specify which individual terms are actually going to be visualized in the workspace. At the bottom of the interface, we tell Maestro exactly how those individual terms should appear. What color, what threshold should be used for hiding term contributions below a certain magnitude, and so on. We can add or remove individual atom types from the combination by control-clicking on them. We're also visualizing each of the cubes occupied by the overall QSAR model, although we could scale this back to the volume occupied ligands in the workspace only using this option here. Right now, the combined terms option is checked, which means that whatever terms are highlighted in the above list are going to be combined in the workspace. You can see what happens as I select subsets. The volume occupied by the cubes changes accordingly. And finally, when we reduce things to just a single term, we can visualize that term's contribution using the color settings defined at the bottom of the interface by turning off the Combine Effects toggle. Let's display all the combined effects again and close this interface. So at this point, we've worked through the process of building a pharmacophore model, and if all has gone well, we can use this pharmacophore hypothesis to screen a database. If you've already created a database, you can click the Search for Matches button to bring up the database screening interface.
So what you do is this model which you have created, okay, on a spatial orientation on the software, now can be compared with a particular compound which is already being there, which is a compound model which is already there in the library. So if both of them are superimposable, that means yes, these two will give rise to that particular output. So it becomes very easy to find out how what can be produced from based on it. Now if it is not there in the database, then that's a new chemical entity, we are getting it formed out. Okay, it's a new chemical which is not being there beforehand, chemical combination being there. So that this system will attribute out in these discoveries being there. So is this thing clear to you guys how QSAR works? I just try to show you how the QSAR works in this part, in portfolio.
Yes, sir.
Okay, beta. So you can search out for QSAR. I'll suggest you, if you want to, there are free videos by Anwar Sheikh. Okay, he's a professor, Professor Anwar Sheikh karke, and his free videos are there on QSAR. He has put across in YouTube. Okay, you can go and see QSAR YouTube videos. Probably you may be able to see it. He's HOD for Alana College in Pune. That's what it is. Another interface. His name is Dr. Anwar Sheikh. Okay.
Okay,
The
sir.
work we had done was in KMK College, KM Kundani College of Pharmacy in Worli Seaface. Okay, that's the place where it is. We had done. So why I'm telling this is because Bida, I know that he is still being he's in academia only for a long time point. I have moved into industry. That's a different thing. So it will give you an idea about how QSR works out, and he has mastered this particular QSR functionality very well. So that's the level one which you would want to know. So it will ease your process of understanding things in terms of doing it actually. So So what happens is it makes you more easy, and you can do things which you take in years, about a decade's time, in maybe 2 hours, 2 days, maximum 15, 20 days, and that helps you to save your time from the nitty-gritties of long-term boring work of continuous observation to anticipatory works is a smart work point of view. And then the next level what we have is AI. AI means artificial intelligence doing it for you, the tedious works. So what you can use is you can use that for drug development process, and that's what is the future in store for you guys. Okay. All right. So I believe this is very clear to you now. So what I'll do is I'll take you further to the last part of this particular 20 seconds more, just to
You can
have.
use the QSAR that you created to screen for matches, although having a QSAR is not a prerequisite for database screening. One final point that I want to remind you of is that you can view useful documentation in your web browser at any time by clicking on the Help button in the lower right-hand corner of any window.
So this was QSAR for you. So I wanted you to have an understanding of QSAR also while we were talking about our critical path talk way of doing it out. So we are on slide number 32, beta. We now need to understand simulation models of drug modeling and simulation studies based on that, designing perspectives which will require us to understand a little bit of statistics and biological biostats point of view. So slide number 32 to 43 I would want to cover in the next presentation because it will be too much for you. As of now I can understand it will be too much of information on your end. So we completed from slide 20 to 32 right now. So 32 onwards I'll have the particular interface in the next lecture. If you are fine with it, beta, we will have the next lecture tomorrow from the same time. Okay, is it fine, beta?
Okay, sir.
Yes, sir.
Yes, sir.
Because
Yes, yes.
otherwise it will be too much for you, beta. Because I've told you so much of information, it's culpably too much for you. So we will have it tomorrow, okay? And I have confirmed with ma'am, so you still have some more time left out for your exam. So we can complete this in the next 2 lectures or so.
Yes, sir.
Okay, I believe everything is clear, beta. Any questions for me before I close the session for today? Any questions?
No, sir.
All clear?
No,
Okay,
sir.
guys.
No, sir.
Yeah, okay, beta. You all have I have the slide deck also, beta. You can go through it also from 33 to 43. We will talk about it in the next one, which will also require you to have an understanding of biostats to a little bit and design strategies, which I would cover in the next lecture. Okay, guys. Thank you so much, beta. All of you. Thank you so much to attend the lecture. We have been running 1.5 hours, and that's normally a good time for understanding enough. Not more than that is to be spent across. So I'll see you soon. That is tomorrow by 11 o'clock. Continuation of from. Till that time, wish
Yes.
you all a goodbye and good day to you. Bye bye, guys.